Hello everyone, welcome to Get My Class. Today we are going to solve this question which says the enzyme required for isolation of protoplast is option A pectinase, option B cellulase, option C both A and B, option D chitinase. So let us understand the question. The question says the enzyme required for isolation of protoplast. So which among these is an enzyme or it is used for isolation of protoplast? So before we look for enzyme, first we have to talk about what is protoplast. So what do you mean by protoplast? Have you heard about this term? Since you have already completed the chapters, you should be knowing protoplast is nothing but the plant cell without the cell wall, without the cell wall, okay? Cell wall. So not only plant cell, even bacterial cell contain cell wall. If you remove the cell wall, what you get is called as a protoplast only. Okay. But then here, since we are talking about the strategies which have been applied in case of plant breeding and plant tissue culture, etc. So that is why we are using this terminology protoplast with respect to plant cell. Okay. So once you remove the cell wall, then it is called as a protoplast. So suppose this is the plant cell. This is the plant cell. And you have the cell membrane and cell wall, right? This is the cell wall and this is the cell membrane. Inner to the cell wall is this is the cell wall and inner to the cell wall is a cell membrane or plasma membrane. Okay, now of course inside you have the nucleus is there, the mitochondria, the chloroplast, ribosomes, everything. Okay, this is a plant cell. So usually plant cells are rectangular in shape. They have different shape. I have just taken this cell. Now, this can be treated with enzymes. Okay. Which type of enzymes? Of course, you have to take the enzyme which can degrade the cell wall. So, what is the cell wall made up of? In plant, the cell wall is made up of what? The major component is what? Cellulose. Cellulose, right? And other component is very important, that is pectin. So cellulose and pectin are the two major components, which are the components, as I said, they are the major components of cell wall. Now, these have to be broken down so as to dissolve the cell wall. And which enzyme will you use? So, of course, to break down cellulose, you have to use cellulase, right? What is cellulase? Cellulase is an enzyme which can bring about the breakdown of cellulase, which can digest cellulase or cellulose. Whereas pectin will be digested by the enzyme called as pectinase. You need the combination of both. Okay, both the enzymes you need. And further, when you give or when you perform this enzymatic digestion of this cell wall from the cell, what you get is the protoplast. Protoplast. And how will the protoplast look? The protoplast look like this. Why? Because the cell wall is not there, only the cell membrane is there. And of course, the rest all remain the same. The cytoplasm, the ribosome, mitochondria, chloroplast, everything will remain the same. Okay, but this is called as protoplast, that is without the cell. Okay, now this can be further utilized for various technologies like which is called as somatic hybridization to produce. I hope you remember about something called as tomato, right? Where the protoplast of potato and tomato could be fused together. Okay, so that's how these protoplasts are useful. Now, let us look at the question again. It says the enzyme required for isolation of protoplasts is option A, pectinase. Yes, pectinase is required. Option B, cellulase. Yes, cellulase is also required because, as I said, then option C says both A and B. So that should be the perfect answer because we need the combination of both. Option D says chitinase. So no chitinase because here as I said, the major component is cellulose and pectin. So chitinase is not required. Okay. So which is the correct answer? So since both the answers, that's A and B are correct, we will go with option C, which consists of both. So let us conclude the answer is option C. I hope you understood the explanation. Thank you, everyone.